COVID reactivated herpes viruses, human endogenous retroviruses, HERVs, and Behçet's disease, Part 2. These are notes for January 29, 2025. In the first part of this multi-part discussion, we discussed these two papers, human herpes virus 6, human herpes virus 7, and Epstein-Barr virus reactivation in pityriasis rosea during COVID-19. This was a reactivation of 3 herpes viruses in response to a COVID infection. And this concept of a primary infection, COVID-2 infection, evoking the reactivation of dormant viruses, this can be considered a sequela or a secondary event to the primary infection. In addition, there is a tertiary reactivation of HERVs, ancient or fossil viruses, and that's abbreviated HERV, HERVs. The second paper we discussed were gum recessions, gingival recessions caused by herpes simplex virus in a patient with COVID-19 infection. So this 3-step process, COVID-2 reactivating a herpes virus, in this case HSV1, and the reactivation of HERVs, leading to the formation of lesions. This shows the complexity of the pathogenesis and helps steer the discussion towards various therapeutic interventions. To help focus this discussion in part 2 of this series, I would like to introduce the condition known as Behçet's disease, also known as Behçet's syndrome. It is an autoimmune disease which expresses itself in recurring severe ulcerations of the oropharyngeal cavity as well as the genital region. In the oral cavity, the oral ulcers of Behçet's often contain reactivated herpes simplex virus 1. And so implication, and this is a hypothesis, that associated with these HSV1 reactivations, there'll be a tertiary reactivation of HERVs that are characteristically activated by HSV1. So we'd like to introduce some of this literature. There are over 14,000 papers on PubMed dealing with Behçet's disease, so it is a very useful set of information to compare with other neurodegenerative disorders, autoimmune disorders, and there are a few papers which have been published on what happens when Behçet's patients contract COVID. We will be discussing those as well. So the first paper I would like to mention, Behçet's disease patients with multiple sclerosis-like features, discrimination value of Barkhoff criteria. Neuro-Behçet's disease misdiagnosed and treated as multiple sclerosis, a deceiving masquerader. Now, as I mentioned in the first part of this series, if you look for HSV1 Alzheimer's, you'll find 325 results. If you look for neuro-Behçet's, you'll find 573 results. So Behçet's, even though it forms visible pathologies, namely ulcerations of the oropharyngeal cavity and the genital region, the pathologies can also be expressed in visceral organs and in the nervous system. So let me just read an AI overview of Behçet's disease. Behçet's disease has a strong genetic link, primarily with the HLA-B51 allele, a gene variant involved in immune response, significantly increasing risk but not causing the disease alone. While HLA-B51 is found in many Behçet's disease patients, especially along the historic Silk Road in the Middle East, Central Asia. It is not diagnostic, as most carriers do not develop Behçet's disease, and other genetic environmental factors are involved. The HLA-B51 gene influences the disease's presentation and severity, often correlating with more severe outcomes, especially in younger males, and interacts with other genes like ERAP1. To better understand how CoV-2 reactivates herpes viruses, particularly HSV1, Behçet's patients may serve as a very important human cohort to understand these reactions. As we mentioned in the first part of this talk, the reactivation of HSV1 in the central nervous system has now been linked as one potential driver of Alzheimer's disease. So the downstream events of HSV1 reactivation involve the reactivation of certain HERVs. And so this sequence of a primary insult, in this case a CoV-2 infection, reactivating HSV1, a secondary response, and the reactivation of HERVs, a tertiary response, leading to pathological lesions, Behçet's ulcers with the presence of HSV1 and potentially the reactivation of HERVs in these lesions should serve as a useful system to understand the impacts, treatments that are designed to suppress long COVID symptoms, the reactivation of herpes viruses. Now, I'd like to tie these concepts together. First, what is the HLA gene complex? It stands for human leukocyte antigen. It's located on chromosome 6. It is essential for immune system function by producing proteins that distinguish self from non-self, presenting antigens to T cells and regulating inflammatory responses. These highly polymorphic genes are critical for defending against pathogens, influencing transplant compatibility and contributing to autoimmune disease susceptibility. And again, Behçet's, with its HLA-B51 allele, predisposes people to this condition, which is viewed as a combination disorder, part autoimmune, part infectious agent. And HSV1 is one of the key infectious agents that's reactivated in Behçet oral ulcers. What about lesions found in neuroBehçet's? On PubMed, there are over 1,200 papers on neurological aspects of Behçet's disease. Here are two of them. Neurological involvement by Behçet's syndrome, clinical features, diagnosis, treatment, and outcome. NeuroBehçet's disease, a review. When diagnosing Behçet's disease, the following international criteria are used. Signs or symptoms are awarded points. Ocular lesions, 2 points. Genital aphthosis, 2 points. Aphthosis is a large, non-healing ulceration, like a canker sore. Genital aphthosis are canker sores in the genital region. Oral aphthosis, oral ulcers, 2 points. Skin lesions, 1 point. Neurological manifestations, 1 point. Vascular manifestations, 1 point. Positive pathergy test. Pathergy is when a bump to the skin results in a inflammation or ulceration. These are the clinical features of Behçet's. However, sophisticated neuroimaging methods like contrasting MRI can detect neurovascular lesions in the brains of certain Behçet's patients. These are then diagnosed as having neuroBehçet's. On the upper left, you see a figure from a paper on neuroBehçet's, showing a neuropathological features of meningioencephalitis of Behçet's. So this is H and E is hematoxylin eosin staining, 60x magnification, showing an infiltration of lymphocytes around a vein. Now, this type of infiltration and aggregation of white blood cells around a blood vessel is known as perivascular cuffing, and the aggregation of white blood cells is called a perivascular cuff. And these are very common in advanced MS patients, very pronounced in large perivascular cuffs. And so this is a sign of hyperimmune state and cells crossing the blood-brain barrier and accumulating between the blood vessel and the basal lamina to which the end feet of astrocytes are attached. This is in the central nervous system. So in the upper right of this slide, I have illustrated a plausible three-step set of events that could produce a perivascular cuffing. One possible scenario by which it could occur in a long COVID patient or a Behçet's patient. COVID-2 would be the primary infection. It reactivates a herpes virus, HSV1. HSV1, in turn, upregulates HERV-K protein, the expression of a HERV virus, an ancient or fossil retrovirus. And this HERV-K protein is immunogenic and pathologic, toxic. The presence of HERV-K protein could lead to a perivascular cuffing. Now, I should mention that perivascular cuffs are not just lymphocytes. They are also filled with leukocytes, including neutrophils. And so, in the discussion of long COVID perivascular spaces, you need to distinguish what types of cells are in these perivascular cuffs. For neurologists and neuropsychiatrists, the presence of persistent perivascular cuffs is a sign of neuroinflammation. Earlier on this YouTube channel, I reviewed the literature on perivascular spaces in the context of long COVID. Persistent and large perivascular spaces, which are detected through MRI, these are part of the lymphatic system, and these 5 YouTubes discuss how infections of the brainstem by viruses, including COVID-2, can result in the enlargement of perivascular spaces. So the perivascular spaces are important from a diagnostic point of view, but also from a mechanistic point of view of understanding neurological disorders such as dysautonomias, which occur as sequelae to the primary viral infections. There are 3 YouTubes on this channel which deal with enlarged perivascular spaces in the context of long COVID. Those are highlighted in green at the bottom of the slide. Now, utilizing AI overview to look at the information about specific HERV virus proteins in the context of neurological disorders. First, looking at multiple sclerosis. Human endogenous retrovirus K, HERV-K, specifically HML2 envelope, and that is abbreviated ENV, and GAG proteins are increasingly recognized as potential pathogenic factors and biomarkers in multiple sclerosis. While HERV-W is more commonly associated with MS, HERV-K expression is also found in MS, with recent sensitive mass spectrometry techniques, often using membrane-enriched samples, detecting these proteins in cell lines and potential links to immune activations. Key finding on HERV-K and MS. Presence in MS. HERV-K GAG and envelope proteins have been detected in MS patient samples. Pathogenic potential. Similar to HERV-W's syncytin-1, HERV-K is believed to trigger inflammatory responses. Now in the upper right of the slide, I have placed a H and E stain, hemotoxin and eosin histological stain of a blood vessel. This was taken as a biopsy from a patient with neurobehcets. And once again, there is an arrangement of white blood cells, in this case leukocytes, surrounding the blood vessel. These are the blue dots. And this aggregation of white blood cells, these are white blood cells that left the bloodstream, crossed the blood vessel, and entered the perivascular space, forming a, quote, perivascular cuff. This is similar to what is seen in multiple sclerosis. This is a conjecture, hypothesis, that the HERV proteins that are secreted in response to the reactivation of dormant viruses, such as herpes simplex virus 1, or other aspects that turn on the promoter of the HERV virus gene, encoding these proteins, whatever is driving the promoters of these retrovirus genes, these ancient fossil viruses, the presence of these proteins activating the immune system, the histological clinical finding could be an enlarged perivascular cuff. That's not to say that all perivascular cuffs would necessarily contain HERV proteins, but in the case of certain neurological disorders such as MS, ALS, Alzheimer's, perhaps long COVID as well, the presence of these HERV-K proteins may be a key element which is promoting the extravasation and accumulation of the white blood cells, both lymphocytes and leukocytes. Utilizing AI overview to look at HERV proteins in the context of amyotrophic lateral sclerosis, ALS. Human endogenous retrovirus K, HERV-K envelope protein, and RNA are detectable in the cerebral spinal fluid of a subset of patients with neurodegenerative diseases, particularly ALS patients, as well as Alzheimer's disease patients. The expression of HERV-K in the central nervous system is linked to neuronal damage, with its envelope protein acting as a neurotoxin that may cause motor neuron degeneration. Key findings on HERV-K in cerebral spinal fluid. ALS pathogenesis. Studies have identified HERV-K HML2 envelope protein and RNA in the cerebral spinal fluid and brain tissues of ALS patients. Neurotoxicity. NERV-K envelope protein is directly neurotoxic, causing the retraction and beating of neurites, projections from nerve cells, in human neuronal cultures. Mechanism of action. The protein induces mitochondrial dysfunction, increases reactive oxygen species, and causes cell death. It is believed to be regulated by TDP-41, a protein often associated with ALS. A recent paper from 2022 is shown at the lower right of the slide. Human endogenous retrovirus K, HERV-K envelope protein in spinal fluid of amyotrophic lateral sclerosis is toxic. So we utilize this slide in the first part of this YouTube series. I'd like to again emphasize that a set of viruses that can provide the primary infections can reactivate these dormant fossil viruses known as human endogenous retroviruses. And when these ancient viruses are reactivated from the human genome, they express proteins, which are frequently toxic and immunogenic. So the key is, what are the transactivators of HERVs in the context of diseases such as long COVID? That will require an analysis of the promoter regions of these ancient viruses. And the metabolic pathways that cells use to reduce reactive oxygen species may be part of the key to understand when HERV viruses reactivate. For instance, under oxidative stress and the excess production of reactive oxygen species, ROS. These pathways, which are promoting cytokine production, may be the triggers for the reactivation of these HERV viruses. And again, would like to emphasize that during viral infections, autoimmune diseases, and cancers, frequently there is a methylation of genes, such as the vitamin D receptors promoter. And there's also the methylation of the GPX4 promoter. And GPX4 is a critical enzyme to deactivate reactive oxygen species as well as peroxidated lipids. And so in cells that have compromised expression of GPX4, this may predispose them to the reactivation of these HERV viruses. And as a consequence of that, a sequela to the reactivation of these HERV viruses, the production of these toxic HERV proteins could initiate changes in cell behavior, which will ultimately manifest in the production of histological lesions such as enlarged perivascular spaces. Now connecting these concepts to COVID infections. This paper from 2023, SARS-CoV-2 awakens ancient retroviral genes and the expression of pro-inflammatory HERV-W envelope protein in COVID-19 patients. Recent studies have identified a significant link between SARS-CoV-2 infection and the activation of human endogenous retroviruses, HERVs, particularly HERV-W envelope protein, ENV, which is suspected to contribute to the severity of COVID and the persistence of long COVID symptoms. A sidebar comment. I would like to again emphasize the potential importance of looking at the reactivation of herpes viruses and HERVs viruses in Behçet's oral ulcers in the context of COVID-2. We will be discussing recent papers where Behçet's patients have been infected with COVID-2 and what happens to their physiology as a consequence. However, in terms of the virology and molecular cellular dynamics of lesion formation, the Behçet oral ulcer may be a very accessible site to study how COVID-2 reactivates HSV1 and then subsequently HSV1 reactivating HERV viruses and their toxic proteins, how all of this may contribute to the persistent ulceration that is found in Behçet's oral ulcers. Now, this is a graphical abstract from the paper we have just been mentioning. SARS-CoV-2 awakens ancient retroviral genes and the expression of pro-inflammatory HERV-W envelope protein in COVID-19 patients. I have condensed what is shown in the graphical abstract of the paper into a linear information vector. COVID-2 reactivating HERV-W directly. There's no herpes virus reactivation involved in this step. Once the HERV-W virus is reactivated, there is expression of the HERV-W envelope protein. And the HERV envelope protein leads to tissue pathogenesis. Now turning to Alzheimer's. Repetitive injury induces phenotypes associated with Alzheimer's disease by reactivating herpes simplex virus 1 in a human brain tissue model. These are organoids. Reading from the abstract. These data suggest that HSV1 in the brain is pivotal in increasing the risk of Alzheimer's disease, as other recent studies using cerebral organoids have suggested. We propose that after brain injury, whether by infection or mechanical damage, the resulting inflammation induces HSV1 reactivation in the brain, leading to the development of Alzheimer's disease/dementia, and that HSV1 is a major cause of the disease, especially in APOE carriers, APOE carriers. This does not preclude the existence of other mechanisms in the etiology, the causes of Alzheimer's disease, particularly those involving other infectious agents. Future studies will investigate possible ways of mitigating or stopping the damage caused by head injury, such as anti-inflammatory and antiviral treatment after injury, thereby preventing HSV1 reactivation in the brain and reducing subsequent development of Alzheimer's disease. A sidebar comment. On this channel, we have been emphasizing that active vitamin D receptor agonists, not vitamin D3, hydroxylated versions of vitamin D, particularly calcifediol 25 vitamin D3 and paracalcitol 125 vitamin D2, used in tandem, could be potentially used to suppress HSV1 reactivation or suppress reactivated HSV1 reactivation in various IAC conditions, or perhaps in the case of traumatic brain injury, TBI, which this paper now suggests is a key driver for the reactivation of HSV1, which may lead to Alzheimer's disease. I would like to draw your attention once again to this paper utilizing active vitamin D hormones, calcifediol, the liver metabolite of vitamin D, which does bind vitamin D receptors and activate them. It's at a concentration of about 1,000 times more concentrated than the kidney metabolite of vitamin D, calcitriol. Calcifediol, its normal reference range is 30 to 100 nanograms per mil. In contrast, calcitriol is about 1,000 times less. Its reference range is 20 to 80 picograms per mil. Calcitriol as a supplement has a side effect that is unwanted. It's deleterious, potentially deleterious. Hypercalcemia. It elevates serum calcium 2 ways, mobilizing calcium release from the bones, as well as increased calcium uptake through the gut. For that reason, nephrologists and endocrinologists have developed an important synthetic analog of calcitriol, paracalcitol, which has much less calcemic effects. For that reason, paracalcitol is now the standard of care for chronic kidney disease patients who lack adequate levels of calcitriol, so they supplement with paracalcitol. This was the motivation for combining these two for the treatment of this patient with severe HSV1 infectious keratitis. The keratitis, the infection and inflammation of the cornea was so severe, the cornea perforated and the iris began to protrude out of the perforated cornea. The patient was hospitalized, treated with standard of care for HSV1 infusions of acyclovir. She was also treated with agents to reduce the osmotic pressure of her vitreous humor to allow the iris to move back inside the corneal boundary. A fetal amniotic chorion membrane was grafted to the cornea. Again, this is standard of care for these types of infections. And she was then started on calcidiol and paracalcitol after she was released from the hospital. While she was being treated, first with a combination of calcidiol and paracalcitol, there was no recurrence of her HSV-1 infectious keratitis, which often occurs in these cases. In addition, the cornea began to heal. At a couple of months after treatment, the paracalcitol was removed, but she was maintained on daily calcidiol. Her cornea began to heal and thicken, but she was left with a corneal scar. You can see that in the right panel. About a year after she was released from the hospital and healed cornea, she had a corneal transplant, and she recovered about 80% normal vision in this eye, a remarkable outcome. So combining this important paper on the use of active vitamin D hormones to treat HSV-1 infection in the cornea with this paper on repetitive injury inducing Alzheimer's by reactivating HSV-1, reading again from the conclusion of this paper, future studies will investigate possible ways of mitigating or stopping the damage caused by head injury, such as anti-inflammatory and antiviral treatment after injury, thereby preventing HSV-1 reactivation in the brain and reducing subsequent development of Alzheimer's disease. So the methods that were used to treat this case of severe infectious HSV-1 keratitis could potentially be translated to patients who have recurring TBIs, or in the case of long COVID, one could conceive that the chronic infections and viral reactivations in the central nervous system of people with long COVID, that again, the use of active vitamin D hormones used in combination could be potentially used as adjunctive therapies with agents like acyclovir to suppress HSV-1 and the associated tissue damage. On this channel, there are a number of YouTubes describing the potent activity of paracalcitol, its ability to suppress ferroptosis. Ferroptosis is a specific form of programmed cell death associated often with viral infections as well as ischemic reperfusion injuries. And both of these pertain to the ontogeny of Alzheimer's disease. So in closing, I would like to summarize the concepts that have been presented in parts 1 and 2 of this multi-component YouTube series. So here is a graphical summary. We're looking at a set of sequential viral transactivations. We can abbreviate that by SVT. First, step 1, a COVID-2 infection reactivates herpes viruses, such as HSV-1 and/or Epstein-Barr virus. These are just two of the herpes viruses that can be reactivated by COVID. Step 3, HERV proteins can be reactivated either by COVID-2, HSV-1, or EBV. These HERV proteins are toxic and immunogenic. So besides the HERV proteins, there are multiple proteins and RNAs being generated by these upstream viral infections. And so what is a result of this is a combined viral pathogenesis, all arriving from a process which we can refer to as sequential viral transactivations. we can utilize the concept of sequential viral transactivations to introduce a key paper which we will examine in more detail in part 3 of this YouTube series. HSV-1 and endogenous retroviruses as risk factors in demyelination. And this paper will be used to analyze demyelination events that are occurring in a subset of long COVID patients. So reading from the abstract of this current paper, herpes simplex virus 1 is a neurotropic alpha herpes virus that can infect the peripheral and central nervous systems, and it has been implicated in demyelinating and neurodegenerative processes. Transposable elements are DNA sequences that can move from one genomic location to another. Transposable elements have been linked to several diseases affecting the central nervous system, including multiple sclerosis, a demyelinating disease of unknown etiology influenced by genetic and environmental factors. Endogenous viral transactivators may activate certain retrotransposons or class 1 transposable elements. In this context, several herpes viruses have been linked to multiple sclerosis, and one of them, HSV-1, herpes simplex virus 1, might act as a risk factor by mediating processes such as molecular mimicry, remyelination, and activity of endogenous retroviruses. Several herpes viruses have been involved in the regulation of HERVs, and HSV-1 in particular can modulate HERVs in cells involved in multiple sclerosis pathogenesis. This review exposes current knowledge about the relationship between HSV-1 and human endogenous retroviruses, focusing on their contribution as a risk factor for MS. Now, looking at the information landscape about virally induced demyelination on PubMed, if you search for demyelination in long COVID, there are about 240 results. In 2025 alone, there are 25 papers on this. HERV and demyelination give you 236 results. 2 key papers. Role of demyelination in the persistence of neurological and mental impairments after COVID-19. Multiple sclerosis and SARS-CoV-2, has the interplay started? Again, looking at this material through the lens of a process, sequential viral transactivation, starting with the COVID-2 infection, reactivation of herpes viruses, reactivation, transactivation of HERV viruses, and the production of toxic, neurotoxic HERV proteins, producing demyelination. And here is one of the figures from the paper we will go into more detail in part 3 of this discussion. Role of HSV-1 in HERV's activation and immune response. The figure summarizes relevant effects of herpes simplex virus 1 on HERV's transcription and the synergistic effects of both viruses on immune response. Again, the concept of combined viral pathogenesis arising from sequential viral transactivation. If you would like a PDF slide summary of this talk, please go to the Google Docs folder highlighted in green at the bottom of the slide. Here you will find a PDF slide summary of this particular YouTube, which contains active links to other YouTubes on this channel, as well as certain references on PubMed. If you navigate to the subfolder Download First, you'll be able to locate this PDF more quickly. At this point, I will pause, and then we will go into part 3 of this discussion.